All living things are made of cells. They are the basic building blocks, if you will, but naturally the closer you look, the more you see. Now this is a generic drawing of an animal cell. It is circular, a bit like a fried egg or an eye. You need to be able to label an animal cell and say what the function or job is for each labelled part. The outer line is called the cell membrane, and it controls the entry and exit of substances in and out of the cell. Think of it a bit like a bouncer outside a club letting certain people in and out. The smaller circle is called the nucleus. The nucleus controls the activities of the cell and contains genetic material called DNA. Some people like to think of the nucleus as the supercomputer of the cell. The space between the cell membrane and the nucleus is called the cytoplasm, and this is where most chemical reactions take place. The smaller tic-tac shaped things in the cytoplasm are called the mitochondria, and this is the site of aerobic respiration. Personally, as a teacher, I don't really like the phrase powerhouse of the cell, so let's stick with this good answer, the site of aerobic respiration. Of course, the structure of a cell in reality is more complicated than this, but this is all you're required to know for your WJEC exam. And remember, this is the board of simplified facts. Now, a good rule of thumb is that a generic plant cell will look more rectangular in shape compared to an animal cell. And it does share some common features with the animal cell, such as the nucleus, the cytoplasm, and the mitochondria, and of course the cell membrane. But notice, there is an extra layer outside the cell membrane called the cell wall. The cell wall is made of cellulose, which provides structural support for the cell. Animal cells do not have cell walls. Another feature of plant cells are these green parts called chloroplasts. They contain a pigment called chlorophyll, and this is where photosynthesis takes place in a plant. So the chloroplasts are the site of photosynthesis. Now remember, this is a generic plant cell. In a later lesson, you will realise that some specialised plant cells don't have chloroplasts, like the ones you'll find below the ground. The larger blob you can see in a plant cell is called the vacuole. It contains a sugary solution called sap. It fills with water and then presses against the cell wall, making the cell firm. A scientific word for this firmness is called turgid. Now, when you don't water a plant, the vacuole will shrink away from the cell walls and cause the plant to wilt. When you rewater the plant, hopefully the vacuoles fill with water again, and your plant starts to look a lot healthier. You should now be able to label and state the function of each part of the animal and plant cell. Taking samples of animal and plant cells and viewing them under a microscope is a specified practical in the WJEC specification. This means that it is very likely a question related to the practical will be in your exam. You will need to be able to describe the step-by-step process of sampling cells and some of the reasons behind some of the steps. Using a cotton wool bud, you first scrape the inside of your cheek, and I stress you must include the word inside. Then you wipe the sampled cells onto the glass slide. The used cotton wool bud is then hygienically disposed of by placing into disinfectant to avoid contamination. Using a pipette, add a couple of drops of methylene blue to the glass slide in order to stain the cells, to make the cell features more visible and so that more detail can be observed. Then, using the mounted needle, you lower the cover slip gently onto the stained sampled cells, which reduces the likelihood of trapping air bubbles between the glass slide and the cover slip. For acquiring a plant cell sample, we use onions. First, we use forceps to peel a thin layer from the inside of a freshly cut onion. A scientific phrase for this thin onion skin layer is called the epidermis. We then place the epidermis onto a glass slide. Using a pipette, we add a couple of drops of iodine solution to stain the plant cells, which should make the cell features more visible and more details can be seen. It is important you say iodine solution, because iodine is a solid when not in a solution at room temperature. Then, using the mounted needle, you lower the cover slip gently onto the stained sampled cells, which reduces the likelihood of trapping air bubbles between the glass slide and the cover slip. Now that we have our slides prepared, we need to view the cells at different magnifications and draw and label the cell features that can be seen using a light microscope. In an exam, you might be asked to label a microscope and demonstrate that you know how to calculate the total magnification being used by the light microscope. What you look through is called the eyepiece lens. Normally this can't be changed, and it makes objects appear 10 times larger. We say the eyepiece lens has a magnification of times 10. The 3 color coded lenses are called the objective lenses, and they can be twisted so we can change which one we use. Each objective lens has a different magnification, for example, times 4, times 10, and times 20. To calculate the total magnification, all we do is multiply the eyepiece lens with the objective lens that is being used. For example, eyepiece lens is times 10 magnification, multiplied with the objective lens with a times 4 magnification, giving us a total magnification of times 40. This specific light microscope can achieve a total magnification of times 40, times 100, and times 200, depending on the objective lens you use. Now the wheel on the side is the focus wheel. We use this to adjust the focus, which is basically how the light hits our eyes so that we can see the cells more clearly. In the specified practical, you are asked to draw and label the cheek and onion cells as they appear under times 40 magnification. Here are some drawings I've produced to show you what you are expected to draw. Notice we only draw and label the nucleus, cytoplasm and cell membrane for the cheek cell because at times 40 you can't see the mitochondria. For the onion cell, the only extra thing you label is the cell wall because again the features like the mitochondria cannot be seen at this magnification. Light microscopes therefore have some limitations as their maximum magnification restricts you from seeing a lot of the smaller structure of cells. Electron microscopes, however, can see smaller structures in better detail as they have a much greater magnification, but they can only view dead tissue. Light microscopes are still able to view living tissues and cells, etc. So, next time you look in a microscope, hopefully you will know how much bigger the microscope is making the object appear. You might get a question in your exam that shows you a picture of a cell and it asks you to work out how many times bigger the picture is compared to the actual size of the cell. Or it can ask you how small the actual cell is compared to the image if it has been magnified by X amount. This video will cover the techniques required to answer these types of questions. Here we have a picture of a cell and the question asks us to measure the diameter. Then it wants us to calculate how much bigger the drawing is compared to the actual size of the cell. And note, it provides us with the actual size of the cell as 10 micrometers. Before we do anything with our ruler, I want to stress the importance of checking our units of measurement because we can run into problems if we overlook them. For example, if a rat is 0.3 meters long and a shark is 300 centimeters long, we can't just say 300 centimeters divided by 0.3 meters, which equals 1,000, because this is telling us that the shark is 1,000 times longer than the rat. This would be wrong. 1,000 rats lined up would overshoot the length of the shark by quite a bit, by 297 meters to be precise. Always do a unit conversion first. I prefer always changing everything into meters. It is a good practice because the unit prefix, centi, milli, micro, etc., represents a meter times 10 to the power of something. For example, for a centimeter, it is times 10 to the negative 2. For a millimeter, it is times 10 to the negative 3, and times 10 to the negative 6 for a micrometer, and so on. So the rat stays at 0.3 meters. But the shark is really 300 times 10 to the negative 2 meters. I know most of you can just convert 300 centimeters into meters with ease, but it is a good habit because you won't find it easy converting megameters into meters, etc. Remember, the centi before meter means to the power negative 2. So we get 300 times 10 to the negative 2 divided by 0.3 meters, which is 10. So the shark is only 10 times longer than the rat. Now don't worry if you are not sure when to divide or multiply. I have a trick for that. Now back to our original question. We look at the units it wants the ruler measurement in. It is in millimeters. We measure the drawing and it is 40 millimeters across. Now it asks us how many times bigger the drawing compared to the actual size of the cell, which is only 10 micrometers across. So we first convert our units so they are all in meters. So 40 millimeters is written as 40 times 10 to the negative 3 meters, and 10 micrometers is written as 10 times 10 to the negative 6. Now, how do we know what goes on top and what goes on the bottom when we're dividing? Here is an equation triangle. The letters in the triangle represent the following. M stands for the magnification, or how many times bigger. The I stands for image size, and the A stands for actual size. So we want to find out M. So we cover the M, and we have I on the numerator on the top, and A as the denominator at the bottom. So image size divided by actual size. So 40 times 10 to the negative 3 meters divided by 10 times 10 to the negative 6 meters, which equals 4,000. So the image is 4,000 times bigger than the actual size of the cell. Let's do another example. We are given another drawing of a cell, and the question gives us the actual size of the cell. It asks us to measure the cell in millimeters again, and then work out how much bigger the drawing is compared to the actual size of the cell. So it's the same as before. It gives the actual size of the cell as 50 micrometers in the question. We measure the picture of the cell, and we find it is 50 millimeters across. Now we need to convert our units into meters. So the drawing is 50 millimeters, which is 50 times 10 to the negative 3 meters, and the actual size of the cell is 50 micrometers, which is 50 times 10 to the negative 6 meters. Then we use our equation triangle, we cover the M, and find we need to use image size divided by actual size. So 50 times 10 to the negative 3 divided by 50 times 10 to the negative 6. We get an answer of 1,000. So the drawing is 1,000 times bigger than the actual size of the cell. A final example. We have an image which we need to measure the diameter of. This is an image of a blood vessel this time. Now we are told this image has been magnified by 20 times. The question asks us to find out what is the actual diameter of the blood vessel in meters. So, measuring the image, we get a measurement of 40 millimeters. I'm then going to convert that unit into meters because it's good practice. So 40 millimeters is 40 times 10 to the negative 3 meters. Now, using the equation triangle, I need to cover the actual size, so the A, and this leaves me with I over M, so image size divided by magnification. So 40 times 10 to the negative 3 divided by 20, which is 2 times 10 to the negative 3 meters. This can also be written as 0.002 meters. Here are some examples of specialized cells that you would find in your body. They share many key features with the generic cell, but they have a few special adaptations. You must remember the following sentences when describing specialized cells. Specialized cells are adapted for specific functions within multicellular organisms. Specialized cells are more efficient in performing specific functions than non-specialized cells. Take a sperm cell. They have a strong tail for swimming the long distance to the egg in the fallopian tube where fertilization normally takes place. You may have a question where they give you a specialized cell and you'll need to suggest how they are adapted for their function or job. By the end of year 10, however, you should have encountered enough different specialized cells that you can recognize some of those adaptations. You will also need to understand the levels of organization within organisms regarding how cells organize themselves to become fully functioning multicellular organisms. You have to learn this order of organization. Let's take a specialized cardiac muscle cell. When you have a collection of these, they form a tissue. Tissues are groups of similar cells with a similar function. Then, with other tissues, they form an organ, and in this case, it is the heart. Organs can be made of many tissues that perform specific functions. The heart needs many different types of connective and muscle tissues working together so that the heart can act as a pump. Organs are then organized into organ systems. So in our example, the heart is part of the circulatory system. The many different organ systems work together to form a functioning organism. Specialized cells, tissues, organs, organ systems to organism. You must remember this order of organization for your exam. The more aware you are of the biology of the human body, the easier this section of the course is. Diffusion is the movement of substances down a concentration gradient. Substances will move from regions where they are in high concentration to regions where they are in low concentration. It is also a passive process, so no extra energy is required for the movement to occur. The net movement of the substance stops once the substance reaches equilibrium. Equilibrium meaning each region has the same concentration of the substance in question. Here is an analogy to help explain the process. If Mr. Pricks passes wind in the corner of a room at a party and releases some sulfuric compounds, they are in a region of high concentration near his backside. The sulfuric compounds, which smell bad by the way, will diffuse to regions where they are in lower concentrations, and the guests at the party will start to smell it. We can track the sulfuric compounds as the guests pass out. The guests closer to Mr. Pricks pass out first, until the sulfuric compounds spread equally around the room and reach equilibrium. The sulfuric compounds are so thinly spread out now that the smell of the fart goes away. You can also imagine some particles that move randomly. If we have two regions where one region has a higher concentration of particles and we can measure the mass of each region, eventually the particles would diffuse and reach equilibrium, and they would have the same mass. Equilibrium has now been reached, and there is no net movement. Remember, movement is still happening, it is just equal in both directions. In the context of biology, diffusion of oxygen and carbon dioxide occurs all the time between cells and the blood. Oxygen and carbon dioxide molecules, or particles, are so small they can fit through the cell membrane pores. Oxygen is usually always in a lower concentration inside the cells compared to the blood on the outside, because the cells use up oxygen during aerobic respiration. The blood is being reoxygenated all the time when it reaches the lungs, so blood oxygen concentration is higher. This means the net movement of oxygen via diffusion is always into the cell. Carbon dioxide is usually always higher in the cells because the cell is producing carbon dioxide as a waste product of aerobic respiration. The blood normally has lower concentrations of carbon dioxide because it is removed from the blood when it reaches the lungs. So, the net movement of carbon dioxide is out of the cell. A practical experiment using whisking tubing can also demonstrate how the cell membrane only allows smaller particles through via diffusion. We will revisit this experiment in more detail in the 1.3 topic on digestion. But simply, if you surround the whisking tubing with water and place glucose, a small molecule, inside the whisking tubing, the glucose will diffuse out into the surrounding water because the glucose particles can fit through the small pores of the whisking tubing. If you put starch, a large molecule, inside the whisking tubing, you will see no starch in the surrounding water because starch molecules are too big to fit through the pores of the whisking tubing. Osmosis is the diffusion of water down a concentration gradient through a selectively permeable membrane. Water will diffuse from a region of high water concentration to a region of low water concentration by a selectively permeable membrane. Net movement of water will stop once both regions have the same water concentration. Water movement is still happening, remember, it is just equal in both directions. We say that equilibrium has been reached. Here are some key points you need to understand regarding osmosis. The highest possible concentration of water is found when you have pure water. When solutes are added to water, like salt for example, they lower the water concentration. So a region of pure water, high water concentration and low solute concentration, will diffuse into a region of salty water, so low water concentration and high solute concentration, via selectively permeable membrane. A selectively permeable membrane is a membrane which allows water molecules through, but not the solute that is dissolved in the water. Without this selectively permeable membrane, osmosis won't occur. Cell membranes act like selectively permeable membranes because water is small enough to pass through the membrane, and additionally, They have special pores to let water through called aquaporins. Don't worry, you only need to know that cell membranes are selectively permeable. If you remember that plant cells have vacuoles that push the cell contents against the cell wall when they are full of water, then if you place a plant cell into water that has a lower water concentration than the inside of the cell, the water will leave the cell via osmosis and cause the vacuole to pull away from the cell wall. The vacuole will refill with water once you place it back into a solution with a higher water concentration than the inside of the cell. We will now look at an experiment that demonstrates what happens when we put plant cells into different concentrations of water. The goal is to find out what is the water solute concentration of the inside of the cell. Measuring the change in mass of potato strips before and after placing them into solutions with different water concentrations, we can get the following results. You can easily get a similar graph in an exam question, but just with a different context. The key is to be able to interpret the results, describe the direction the water has moved in, and be ready to use all the correct terminology regarding osmosis. If the potato mass has gone up after being in the solution, then the solution has a higher water concentration compared to the inside of the potato cells. The water has moved into the potato cells across the selectively permeable membrane of the cells in a process called osmosis. The scientific term for a solution that has a higher water concentration than the inside of the cell is a hypotonic solution. If the mass of the potato strip has gone down, then the solution has a lower water concentration compared to the inside of the potato cells. The water has moved out of the potato cells across the selectively permeable membrane of the potato cells via a process called osmosis. The scientific term for a solution that has a lower water concentration than the inside of the cell is a hypertonic solution. If the mass has not changed, then this means no net movement of water has occurred across the selectively permeable membrane of the cell via osmosis, and therefore the system is in equilibrium. This must mean that the solution has the same water concentration as the inside of the potato cells, and a scientific term for this type of solution is called isotonic. On a side note regarding animal cells, because they have no cell wall, it means placing an animal cell into a hypotonic solution, like pure water, can cause the cell to burst. Active transport is the movement of substances against a concentration gradient. Substances are actively transported from a region of low concentration to a region of high concentration. This process requires energy in the form of ATP, which is provided when cells aerobically respire. A classic example in biology where active transport takes place is in the root hair cells of plants. They will absorb solutes such as mineral ions against the concentration gradient, with the goal being to lower the root hair cell water concentration. This ensures that the process of osmosis continues effectively by preventing equilibrium being reached between the soil and the root hair cells. Root hair cells also have lots of mitochondria, which release energy in the form of ATP via aerobic respiration to drive the active transport. A common question that pupils lose marks on is failing to link that active transport relies on aerobic respiration. For example, a question might show some data regarding water uptake by a plant in different conditions. You might be asked to explain why the rate of water uptake is lower when the plant has been poisoned and can no longer utilize oxygen. Now, of course, because there is no oxygen, that means aerobic respiration cannot take place. This means no energy, ATP, is being released, and active transport requires an input of energy in the form of ATP. This means the root hair cell is unable to absorb mineral ions against the concentration gradient, and the water concentration inside the root hair cell will reach equilibrium with the soil the plant is in. The rate of water uptake is therefore reduced. Enzymes are proteins that are made by cells that are involved in all metabolic reactions by breaking down larger molecules into smaller ones or building larger molecules from smaller ones. They are often called biological catalysts because they speed up the rate of chemical reactions. There are many different types of enzymes that do many different jobs. Enzymes are proteins and are therefore made of amino acids. If we take amylase, which is an enzyme that breaks down starch, it will be made of many different amino acids that form a chain which then folds into a specific shape. Think of amino acids as Lego bricks. You have different Lego bricks as you have different amino acids. Then, depending on how you put the Lego bricks, the amino acids, together, you get a different enzyme. The special power amino acids have that Lego bricks don't have is that amino acids can fold, bend, and twist. The specific order of amino acids is hugely important. You change the sequence, you change the enzyme structure, and therefore change its job. Enzymes have a specific shape, and the most important part is the active site. Amylase has a specific site that only lets starch fit into it, and when the starch fits into the active site, it's broken down into smaller molecules such as glucose. The lock and key theory describes how enzymes behave like a lock. The substrate is a specific key, glucose in this case, that fits the enzyme's lock, amylase in this case. They fit together to form an enzyme-substrate complex. Then, a reaction takes place which breaks down the starch into glucose. Enzymes and substrates need to collide in just the right way so the substrate binds to the active site. Temperature describes how quickly particles, such as molecules, move. The higher the temperature, the more the molecules vibrate, and the more kinetic energy they have. Kinetic energy describes motion in this case. Higher temperature, higher kinetic energy, therefore more motion. The idea is that the higher the temperature, the faster molecules move, and the greater chance they have of colliding. So enzymes work faster at higher temperatures because they collide more. The problem is, the enzyme protein structure is not indestructible. If temperatures get too high, then the molecules vibrate more and more until it loses its shape, and the protein unravels. The active site's specific shape is lost, and the substrate can no longer fit into the enzyme's active site. If you change the lock, the old key no longer fits. We describe an enzyme that has lost the specific shape of its active site as denatured. If we graph enzyme activity against temperature, we often see this pattern. At low temperatures, the enzymes and substrates have very little kinetic energy, so there are less collisions happening. The enzyme activity is low. The shape of the active site, however, is unaffected. A bear keeps its shape when it freezes to death. As the temperature increases, the rate of collisions, as the enzyme and substrate gain more kinetic energy, continue to grow. This continues until the activity peaks and the enzyme is at the optimum temperature. At this peak, the enzyme keeps its shape and is making as many collisions as possible. However, we increase the temperature a bit further and the activity drops sharply. The kinetic energy is too high and the enzyme vibrates too much until it loses its structure. The active site's specific shape is lost and the enzyme is denatured. Boiling enzymes, for example, is a common way in practical work to denature enzymes as a control experiment when looking at enzyme activity. pH can also affect the rate of enzyme activity. This graph shows that the enzyme has an optimum pH of 2. This is where the enzyme activity is at its highest. A pH that is too high or too low can denature an enzyme. This WJEC specified practical investigates what is the optimal temperature of the enzyme amylase. We will go through some potential questions they could ask in an exam and the scientific terms you should be using when discussing this practical investigation. We won't go through the method, but the illustration should help remind you of how the practical is conducted. It is important that whatever investigation you have in front of you, that you can identify all the variables. In this investigation, we will be changing the temperature that the enzymes are exposed to. So, this is our independent variable. Our dependent variable will be measuring the color change of a starch test over time. The control variables are the volumes and concentrations of starch and amylase that we use. Also, the time intervals when an observation is recorded regarding color change should be kept the same. A common question in an exam can be to identify a control experiment that we can do to show it is the enzyme that is affecting the color change. In this case, we can boil the amylase, which will denature them, showing that it is the enzymes that is causing the color change and breaking down the starch into glucose. Another thing to consider is what are some good practices we can do regarding collecting data. Where possible, increase the repeatability by doing the experiment 3 times for each temperature. Note, if the range between repeats is small, then this also suggests our data is repeatable and it demonstrates precision. We can then calculate a mean. If other groups or classes get similar data, then we can also say our data is reproducible. If you are not sure how to explain the biology behind these data, then please watch the previous video on enzymes. So, the results show that 40 degrees Celsius is the optimum temperature for this enzyme, as it took the least amount of time to break down all the starch into glucose and produce a negative starch test. You could be asked about the resolution of the thermometer, which in most cases has intervals of 1 degree Celsius, so the resolution is plus or minus 1 degree Celsius. It is also worth noting that we can't be sure if 40 degrees Celsius is the optimum, as it could be a temperature between 30 and 40 or 40 and 50 degrees Celsius. So, some further work we could do would be to look at changing the temperature intervals down to every 5 degrees and looking at a range of temperatures of 30 degrees Celsius, 35, 40, 45 and 50 degrees Celsius to try get closer to the true value of the optimum temperature. Aerobic respiration is an enzyme-controlled process that happens in the mitochondria. Glucose reacts with oxygen and energy is released in the form of ATP. Water and carbon dioxide are also produced as byproducts of the reaction. You need to learn the word equation for your exam. No excuses. In chemistry, there is a process called combustion, which is essentially identical to aerobic respiration. The difference is, aerobic respiration is a more controlled reaction. We don't just set fire to glucose inside our bodies and create an out-of-control chain reaction. There is an experiment involving germinating peas which demonstrates that aerobic respiration releases heat energy. So, it is an exothermic reaction. Just a quick look into germination. Peas are seeds. Seeds have stored energy in the form of starch. Depending on the plant species, different conditions will trigger a seed to germinate. Whiskey is made by letting barley seeds be exposed to perfect conditions to trigger germination. The barley seeds are tricked into germinating and they produce amylase. This breaks down the stored starch into maltose. You might have heard the phrase, malt whiskey. It was at this moment that Mr. Pricks remembered that alcohol is flammable. Back to our germinating peas experiment, we have the following setup. We have some living peas and some dead peas. The dead peas are normally boiled and act as a control experiment and demonstrate that it is the living peas aerobically respiring causing the temperature rise. To record the temperature, we have a thermometer. The thermos flask and cotton wool are used to help reduce heat loss. The cotton wool also lets oxygen into the flask. The disinfectant is used to kill any microorganisms like bacteria. The bacteria, if not killed, can begin using the glucose released by the peas to aerobically respire themselves, causing the temperature to rise and affect the results. This experiment's results show a temperature rise over time as the peas aerobically respire. Eventually, the temperature rise stops because they run out of glucose. The boiled peas should not cause a temperature change. I'm going to present a tricky version of this experiment to try and prepare you for the exam. So we now have a setup where we have living peas with strong disinfectant, boiled peas, boiled peas with weak disinfectant, and boiled peas with strong disinfectant. We see the following temperature changes over time. In living peas, we see the fastest temperature rise because the bacteria are also aerobically respiring. In the living peas with strong disinfectant, we see a temperature rise, but it peaks later because only the peas are aerobically respiring. In the boiled peas, we see a slower temperature rise, as only the bacteria are aerobically respiring, using the peas as an energy source. In the boiled peas with weak disinfectant, we see an even slower temperature rise, as a few surviving bacteria gradually multiply and aerobically respire. In the boiled peas with strong disinfectant, there is no temperature rise because there are no living microorganisms to aerobically respire. No aerobic respiration is taking place. Anaerobic respiration is a process where glucose is broken down into lactic acid and energy is released in the form of ATP. Learn the equation. Notice the process doesn't need oxygen, but sadly, we can't do this for very long. Aerobic respiration is for everyday activities and low-intensity exercise like jogging. Anaerobic respiration is used for high-intensity exercise such as sprinting. Muscles need energy. That energy comes normally from ATP that is made during aerobic respiration. Remember that cells need a constant supply of oxygen from the blood to complete aerobic respiration. When we exercise vigorously, our muscles demand more energy because they are working harder. So in theory, they need more oxygen reacting with glucose via aerobic respiration. The trouble is, your body can't supply oxygen fast enough to the cells to meet the energy demand if the exercise is too vigorous. We know from experience that during exercise, our breathing rate increases, as does our heart rate, in an effort to supply the cells with enough oxygen when we exercise. The muscle cells are demanding more energy, but the oxygen isn't coming in fast enough. So, they start to break down the glucose into lactic acid, which releases some ATP. The problem is, lactic acid is harmful to the body. It causes muscle cramp and weakness. That means the body has to break it down at some point. The trouble is, you need oxygen to do that. So at some point, your body gives up exercising because the lactic acid buildup is too high. Once you stop exercising, the body still needs to use up extra oxygen to break down the lactic acid. And we call this an oxygen debt. You will have noticed that after a sprint, you are still breathing heavily and your heart rate is high, as the body still needs to get lots of oxygen into the blood and deliver it quickly to the cells. That extra oxygen is needed to break down the lactic acid. For higher tier, you also need to know that anaerobic respiration is less efficient than aerobic respiration because of the incomplete breakdown of glucose. Less ATP is produced per molecule of glucose in anaerobic respiration than in aerobic respiration. Exam questions on this topic are answered poorly because you normally have to interpret awkward-looking data into strong exam-friendly answers. This data shows us the heart rate and the lactic acid in blood of an athlete before, during, and after a 200-meter sprint. The race started at the 3-minute mark and lasted around 25 seconds. We need to explain the data using our knowledge of anaerobic respiration and oxygen debt. Prior to the race starting, we can identify what a normal level of lactic acid is on the graph. Then, at the 3-minute mark, as the race starts, we see a rise in lactic acid in the blood. Between 4 minutes and 7 minutes, we see a gradual decline in lactic acid levels in the blood. The athlete had an oxygen debt that lasted 4 minutes, as lactic acid levels in the blood did not return to normal until the 7-minute mark. The heart rate is higher during the period of exercise because the muscle cells require more oxygen to be supplied to them by the blood so they can aerobically respire more. If there is not enough oxygen supplied to the muscle cells, then anaerobic respiration starts to take place. The heart rate remains high after the race because extra oxygen is required to break down the excess lactic acid and repay the oxygen debt. More blood needs to be supplied to the cells to repay the oxygen debt. The heart rate returns to a resting rate once the lactic acid has been broken down to normal levels. Who is fitter? Often in an exam, you must interpret graphical data and apply your knowledge about aerobic and anaerobic respiration to determine who is fitter and why. But first, some important points. In general, fitter people show the following traits regarding exercise. Their heart rate is lower during exercise. Their heart rate recovers faster after exercise. They don't produce as much lactic acid during exercise. They can repay oxygen debts faster after exercise. They can inhale a larger volume of air into their lungs compared to unfit people. They are more efficient at utilizing oxygen, so they rely less on anaerobic respiration. This can show up as more carbon dioxide being produced because they are aerobically respiring more. If you can remember these general ideas about fitness and apply some common sense to exam questions, you should be okay. The hardest type of question is probably looking at carbon dioxide output. Pupils sometimes struggle with the concept that more carbon dioxide being produced can be a good thing. If you are producing lots of carbon dioxide during an intense workout compared to somebody else, that means you are doing more aerobic respiration. Those that show a lower carbon dioxide output are relying more on anaerobic respiration. Imagine 4 individuals on an exercise bike and they all must cycle at the same intensity. On modern exercise bikes, the energy you put into pedaling can be measured in joules per second, or watts. So, they must all try and ride at an output of 350 watts of energy, which is hard work by the way. We will look at carbon dioxide levels produced over time, and the data also shows us when riders stop due to cramp in the legs. Notice that individuals who have higher levels of carbon dioxide produced last longer on the bikes. This is because they are aerobically respiring more. Remember, carbon dioxide is a product of aerobic respiration. Those that are producing less carbon dioxide are anaerobically respiring more and producing more lactic acid, and that is why they stop pedaling first. Remember, they are all expending the same amount of energy at 350 watts. The trick is to look at how they are getting that 350 watts of energy. Single-celled organisms, like amoeba, can rely on simple diffusion to meet their oxygen demands. The oxygen doesn't need to travel far to reach every part of the amoeba. Complex multicellular life needs a more complex respiratory system, so they can obtain a large enough volume of oxygen to maintain optimal levels of aerobic respiration. They also need to remove waste gases such as carbon dioxide quickly. Simple diffusion of oxygen through the human skin would result in death. There isn't enough surface area available on the outside, so life has adapted to having structures, like lungs, which have a large surface area, on the inside. Complex multicellular organisms also have a delivery system, the blood in most cases, to make sure the oxygen gets to every cell in the body. It is important to understand that the respiratory system in complex organisms like humans has evolved to ensure efficient gas exchange. You must be able to label the respiratory system in humans. Starting at the top, we have the nasal cavity, where air enters the system. Ideally, we breathe through our noses to warm the air before entering the lungs to reduce irritation. Also, the nose hairs prevent some harmful particulates from entering the lungs. Then, the air travels down the trachea, which is in front of the esophagus. Remember, you have 2 tubes, and I'm sure you've experienced food going down the wrong pipe and you start coughing. The trachea also has rings of cartilage to keep the structure open but flexible. Then, the air can go down into either the left or right lung via the bronchi. The bronchi then branch out many times, and these smaller branches are called the bronchioles. At the end of the bronchioles are the alveoli. This is where gas exchange occurs between the blood and the alveoli. Obviously, you need to be able to label the lungs themselves, and surrounding the lungs are the ribs. Between the ribs are the intercostal muscles, which cause the ribs to expand and move up, and contract and move down. Finally, underneath the lungs is the diaphragm, which is the muscle that controls inspiration and expiration. The lungs are also adapted to keep foreign objects and microorganisms from entering them. Cilia line the bronchial walls and are hair-like structures which can flick back and forth. Mucus is produced by the lungs to trap dirt particles like dust and potential harmful microbes, and then the cilia push the mucus out of the lungs and to the back of the throat. it is much easier to cough up mucus that has been forced to the upper areas by your cilia. When we breathe in, inspiration, the diaphragm contracts and flattens. The intercostal muscles contract and pull the ribs upwards and outwards. The volume of the thoracic cavity increases and the pressure inside the lungs decreases. The atmospheric pressure is higher than the pressure in the lungs. Air rushes into the lungs down a pressure gradient. When we breathe out, expiration, the diaphragm relaxes and domes upwards. The intercostal muscles relax and pulls the ribs downwards and inwards. The volume of the thoracic cavity decreases and the pressure inside the lungs increases. The air pressure inside the lungs is higher than the atmospheric pressure. Air rushes out of the lungs down a pressure gradient. Remember, when we breathe in, all the muscles just mentioned contract. When we breathe out, all the muscles mentioned relax. Now on to the next half of the video, looking at this in more detail. You won't need the following information to answer exam questions, but if you understand the process more, then you should be able to remember how the process works. Everything I mention will be simplified, and there won't be too much terminology. Pressure measures the collective force when particles collide on the walls of a container. If you have the same number of particles and the temperature is constant, then if we increase the volume, the pressure decreases. The collective outward force felt on the walls of the container is less. If we shrink the container and reduce the volume, the pressure goes up as the collective force felt on the container increases. The atmosphere is constantly applying pressure on our bodies, and given a chance, the atmosphere will enter our lungs. You can open your mouth, however, and notice that air doesn't rush in and inflate your lungs. This is because the pressure inside the lungs is equal to the atmospheric pressure, so the body needs to do more than just open the mouth. The lungs are surrounded by a membrane folded over itself to make a space filled with fluid. It is connected to the lungs and the thoracic walls. Fun fact is that the pressure in that space is less than atmospheric pressure, so the lungs are always a bit inflated. If you get a puncture in the cavity, it can cause the lung to collapse. This is because the lungs have elastic walls that want to collapse and cause the lungs to reduce in volume, combined with the increase in pressure surrounding the lungs. The amount of fluid in that space is always kept the same, so if the volume changes, the pressure changes. When the intercostal muscles contract and pull the ribs up and out, it pulls the outer layer of the membrane with it, increasing the volume of the space surrounding the lungs and therefore decreasing the pressure. The membrane that is touching the lungs is pulled outwards, and so are the lungs. So the lung volume increases, and the pressure decreases, so air rushes in. The diaphragm itself contracting also increases the thoracic cavity volume, causing the same effect. When we exhale, the muscles involved relax, and because the rib cage can recoil back and the elasticity of the lungs wanting to pinch back inwards, it decreases the volume of the lungs. This combined with the diaphragm doming upwards will cause the thoracic cavity volume to decrease. This forces air out of the lungs and into the atmosphere because the pressure is higher in the lungs compared to the atmosphere. Here is a pomelo fruit, and they kind of look like lungs. If we squeeze the fruit, the volume decreases and the pressure increases, causing the juice to be forced out. The bell jar model is a great way to demonstrate how the diaphragm contracts and relaxes, resulting in the lungs inflating and deflating. The model does have some limitations, and we will go over those in this video. The tube entering the bell jar represents the trachea, which then splits into the bronchi. The balloons represent the lungs. The bell jar casing represents the rib cage. The rubber sheet represents the diaphragm. The key thing is to realise the bell jar is airtight, and so the amount of air in that space is always the same. The air particles in that jar are pressing not only on the bell jar walls, but also the balloons themselves. So, when we push the rubber band up to mimic the diaphragm relaxing and doming upwards, it decreases the volume of the bell jar and increases the air pressure. The air particles have less space to move around in, so they exert more pressure on the outside of the balloon, causing them to deflate. When we pull down on the rubber sheet, mimicking the diaphragm contracting and flattening, we increase the volume of the bell jar cavity and decrease the pressure. The air particles have more space to move around, so they exert less force on the outside of the balloons. The atmospheric pressure is greater than the pressure in the glass jar and inside the balloons, so air rushes into the balloons. The limitations of the model are as follows. The bell jar is rigid, unlike the rib cage. We can't simulate the intercostal muscles contracting and relaxing, causing the rib cage to move up and outwards, and downwards and inwards. The diaphragm flattens when it contracts, but in the model, we must pull the rubber sheet down quite a lot. The bell jar is full of air, but in reality, the thoracic cavity is filled with bodily fluids. The bell jar has large spaces around the balloons, but again, in reality, the space around the lungs is small in the thoracic cavity. The alveoli are the site of gas exchange between the atmosphere and the blood. Deoxygenated blood flows all around the alveoli so that oxygen can be picked up and carbon dioxide can be removed via diffusion. Oxygen is always in a higher concentration in the alveoli because it is always receiving fresh atmosphere as we breathe. The deoxygenated blood will always have a lower concentration of oxygen compared to the alveoli because the cells have been aerobically respiring and using up the oxygen. Notice the cells produce carbon dioxide, so the blood always has a higher concentration of carbon dioxide compared to the alveoli. You need to be able to label the alveoli. The tube leading into the alveoli is a bronchiole. The wall of the alveolus has a thin, moist layer. The capillaries, which are blood vessels, surround the alveolus. The capillary wall is 1 cell thick. The blood flowing through the capillaries has red blood cells and blood plasma. The red blood cells can carry oxygen and carbon dioxide because they have a special molecule called hemoglobin. But some carbon dioxide is also found dissolved in the blood plasma. On a diagram, you will need to be able to add arrows showing the direction that oxygen and carbon dioxide are diffusing to. Oxygen is always moving into the blood via diffusion. Carbon dioxide is always moving into the alveoli via diffusion. The body needs efficient gas exchange to take place. The alveoli are adapted to facilitate this. They have a special shape which provides a large surface area so more efficient gas exchange can occur. The alveolar wall is thin to reduce the diffusion distance and allow for more efficient gas exchange. The lining is moist, so gases like oxygen and carbon dioxide dissolve and can diffuse quicker through the alveolar wall. The alveoli have a rich blood supply because they're surrounded by capillaries to maximize the rate of diffusion of oxygen and carbon dioxide. The capillaries themselves have a wall that is 1 cell thick, again to maximize the rate of diffusion by reducing the diffusion distance. You must learn the gas percentage composition of inhaled air versus exhaled air. Inhaled air is just the atmosphere, of course. 78% of the atmosphere is nitrogen, 21% is oxygen, and 0.04% is carbon dioxide. The water vapor content varies depending on humidity. The composition of exhaled air is as follows. Nitrogen doesn't change, so is 78%. This is because nitrogen is an inert gas, and our body can't use it. Oxygen drops to 16% because the body has used up some oxygen for aerobic respiration. Notice our bodies don't use up all 21% of the oxygen because they're just not that efficient. Carbon dioxide levels increase to 4% in exhaled air because carbon dioxide is produced by aerobic respiration. Water vapor is saturated in exhaled air because the alveoli are moist and the air is warmer so it can hold more moisture. A simple huff and puff demonstration can show how exhaled air has more carbon dioxide than inhaled air. Lime water is used as a test for carbon dioxide. If the lime water turns cloudy, or some describe it as milky, then that means carbon dioxide is present. Inhaled air is drawn through one boiling tube containing lime water, and then exhaled air is passed through a second boiling tube containing lime water. The exhaled air should turn the lime water milky. Smoking negatively affects the respiratory system in many ways, but you must know the following for your exam. Tar in cigarette smoke contains carcinogens, which can lead to lung cancer and potentially death. A carcinogenic substance causes cancer. A common question that looks at data of the amount somebody smokes versus lung cancer rate will ask why the following statement is incorrect. Smoking is the only cause of lung cancer. You should refer to the data in your answer, which tells us that some people still develop lung cancer even though they don't smoke any cigarettes. Other data interpretation exam questions will ask you to describe trends regarding average cigarettes smoked by a population per year and lung cancer rates. You should notice that there is a lag phase between when smoking increases and when lung cancer rates increase. Cigarettes also contain nicotine, which is the chemical that leads to somebody becoming addicted to smoking. Smoking can also cause emphysema, which causes shortness of breath. This is because cigarette smoke can cause the alveoli walls to weaken and burst. This reduces the surface area of the lungs and leads to inefficient gas exchange. Patients with emphysema may rely on breathing in pure oxygen to help meet their oxygen requirements. Cigarettes also contain chemicals which can paralyze the cilia, so they can no longer move mucus up and out of the lungs. This causes mucus to build up, and the particles in cigarette smoke also make the mucus drier and thicker and clog up the bronchioles. Bacteria can take advantage of this and reproduce by using the mucus as a food source. This increases the risk of getting an infection in the lungs. Smoking has many more negative effects, and WJEC will accept a wide range of answers on smoking. But what we have covered in this video are the most commonly accepted answers on smoking. We need digestion because food contains large insoluble molecules that can't dissolve in the blood or be absorbed by the cells. Digestion breaks down large insoluble molecules into smaller soluble molecules. You must learn the following large insoluble molecules and their soluble products. Fat is broken down into fatty acids and glycerol via digestion. Proteins are broken down into amino acids via digestion. Starch is a carbohydrate. It is made of long chains of glucose molecules. Digestion breaks down the starch, separating the chains of glucose molecules into separate individual glucose molecules. Enzymes are involved in the process of digesting all of the mentioned molecules. You must also learn the food tests, which can tell you if you have starch, protein, or glucose in your food. We use iodine solution to test for the presence of starch. A positive result for starch would mean the iodine changes from brown to blue-black. Burette solution is used to test for the presence of proteins. A positive result for protein would cause the burette solution to change from blue to violet. Finally, Benedict's reagent tests for the presence of glucose. This test requires that you boil the solution in a water bath, whereas the previous ones just required adding the solution to the food item. A positive result for glucose would cause the Benedict's reagent to change from blue to brick red. You need to learn the names of the enzymes that break down fats, proteins, and starch, and where you would find them in the digestive system. Amylase, sometimes called carbohydrase, helps to digest starch. Starch is made of long chains of glucose molecules. It might be helpful to think of enzymes as biological scissors that can cut bonds and break molecules apart. Amylase can be found in the saliva in the mouth, so starch digestion starts in the mouth. If you chew bread for long enough and don't swallow it, you will end up with sugars like glucose in your mouth, and the bread could start to taste sweeter. Amylase is also found in the small intestine and is secreted by the pancreas, where again starch is broken down. Amylase can't operate in the stomach because the pH is too low and is denatured in the stomach, so no starch is broken down in the stomach. Protease helps to break down proteins into amino acids. Proteases can be found in the stomach, where they can still perform in the low pH conditions. Proteases are also secreted by the pancreas into the small intestine. Lipase helps to break down fats into fatty acids and glycerol. Lipase is secreted by the pancreas into the small intestine. The pH in the small intestine is between 7 to 8 pH. And again, lipase is not found in the stomach, as lipases would become denatured in the stomach. So, starch is broken down by amylase in the mouth and the small intestine. Proteins are broken down by protease in the stomach and small intestine. Fats are broken down by lipase in the small intestine. You need to be able to label each section of the digestive system and describe the role they play in digestion. Starting with the mouth, food enters here and is chewed into smaller pieces, which increases the surface area exposed to enzymes. We call this physical breaking up of food mechanical digestion. Starch digestion also starts in the mouth, as amylase is found in saliva. When enzymes break large insoluble food molecules into smaller soluble molecules, we call this process chemical digestion. The food is then swallowed and travels down the esophagus. The esophagus has muscles which contract behind the food and relax ahead of it. This forces food down the esophagus in a process called peristalsis, which happens throughout the digestive system. I like to imagine it like squeezing toothpaste out of a tube. The food then enters the stomach, where proteases are secreted, so proteins are broken down here. The amylase from the saliva, if it makes it to the stomach, denatures, because the pH in the stomach is around 1 to 2, due to the presence of hydrochloric acid. Then the food enters the small intestine where carbohydrates, proteins, and fats are broken down. The pancreas secretes into the small intestine proteases, carbohydrates, and lipases. The chemically digested soluble food molecules are then absorbed into the bloodstream via the small intestine. We'll cover absorption more in a later video. The liver secretes bile into the gallbladder. The gallbladder acts as a reservoir ready to release bile into the small intestine via the bile duct when food enters the small intestine. We will cover the role bile plays in a later video. The large intestine carries the undigested waste and fibre to the rectum. Water is also reabsorbed in the large intestine to help solidify the feces. The rectum holds the feces until it is full, which then signals the brain that you need to go to the toilet. The anus is the exit and is the final part of the digestive system. Oh! Oh my god, oh my god. Run. Oh, I'm gonna hide in here. Oh, toilet. Close the door. Maybe he won't find me here. Well, maybe his rectum was full. You gotta go. Huh? Bile has two primary functions. It raises the pH of the acidic food that has just left the stomach, and it emulsifies fat. The stomach contains hydrochloric acid, and eventually the food that is being digested there needs to move on to the small intestine. The problem is the small intestine is not adapted for holding acid, so the acid needs to be neutralized. Bile is alkaline, so when food enters the small intestine, the gallbladder releases the bile. The bile travels through the bile duct and mixes with the partially digested food from the stomach. Lipases, carbohydrates, and certain proteases have an optimum pH of 7 to 8, so the bile generates the favourable conditions in the small intestine. Bile also emulsifies fat. Emulsification describes how the bile emulsifies large droplets of fat into small droplets of fat. This increases the available surface area for lipase enzymic action. Fat digestion is faster when bile is present. This experiment shows how emulsification can decrease the time it takes for fat to be digested by using milk, sodium carbonate, a pH indicator called phenolphthalein, and washing up liquid. Washing up liquid contains surfactants which emulsify the fat in the milk, and this will mimic what bile does. We will have two test tubes that each contain milk, sodium carbonate to make it alkaline, the phenolphthalein indicator, but only one test tube will contain washing up liquid. Phenolphthalein turns pink in an alkaline solution and will go colourless when in neutral to acidic conditions. When we add lipase to both test tubes and observe which one will go colourless first, we should notice that the test tube that has washing up liquid goes colourless first, followed by the second test tube without washing up liquid. The reason we see a colour change at all is because lipase is breaking down the fats into fatty acids and glycerol. The fatty acids that are produced are making the solution more acidic. The reason the test tube with washing up liquid changes colour first is because the washing up liquid has emulsified the large fat droplets into smaller fat droplets, which increases the available surface area for lipase action. Visking tubing can be used to model the small intestine regarding absorption. We will look at how the small intestine is adapted for absorption and the limitations the visking tubing has as a model of the small intestine. In our small intestine, the large insoluble food molecules, starch, protein, and lipids, are being broken down into their respective smaller soluble food molecules, glucose, amino acids, and fatty acids plus glycerol. Only the smaller soluble molecules can diffuse through the small intestinal wall and into the bloodstream by diffusion. The wall of the small intestine acts like a semi-permeable membrane. The small intestine has the following adaptations for efficient absorption. It has a large surface area, which is generated by the special projections the small intestinal wall has, called villi. They also have a rich blood supply, which maintains a steep diffusion gradient. Take glucose, for example. The glucose is diffusing from a region of high concentration, the inside of the small intestine, to a region of low concentration, the blood. If no fresh blood is passing through the villi, eventually the glucose would reach equilibrium and the net movement of glucose would be equal in both directions. Luckily, our heart pumps fresh blood all the time, so glucose is always carried away from the villi, keeping the glucose concentration always low in the villi. Visking tubing can model the small intestine, and exam questions can catch pupils out on this topic as there are many different things they could ask you. Let's put inside the visking tubing some starch, some amylase, and some protein, and then place that now filled Visking tubing into a beaker with some pure water. We will test the water immediately for starch, glucose and protein using the food tests. Remember, we use iodine solution to test for starch, we use Benedict's reagent to test for glucose, and burette solution to test for protein. All the tests should come up as negative, as there's been no time for anything to diffuse into the pure water yet. Now, after every minute, we will test the water for starch, glucose and protein. We then get the following results, which we will now explain. The starch test is negative each time, because the starch molecules are too large to fit through the pores of the semi-permeable membrane of the Visking tubing. The test for protein also remains negative each time, because again, the protein molecules are too large to fit through the pores of the semi-permeable membrane of the Visking tubing. Finally, the glucose test shows stronger and stronger positives as the time passes. Remember, we never put glucose inside the Visking tubing. However, the amylase has been breaking down the starch into glucose. The glucose molecules are small enough to diffuse through the pores of the semi-permeable membrane of the Visking tubing. This takes us to the limitations. The Visking tubing represents the small intestine, but unlike the small intestine, the Visking tubing has a much smaller surface area as it does not have villi, so the rate of diffusion in theory will be much slower. The other limitation is that water is supposed to be representing the blood, but as there is no fresh water entering the beaker, we reach equilibrium and there is no diffusion gradient, and therefore there is no net movement of glucose in either direction. In our bodies, fresh blood is always being supplied and surrounding the small intestine, so the diffusion gradient always remains steep and equilibrium is not reached. This video will tell you exactly what you need to know for your WJEC exam regarding a balanced diet and the fate of fats, carbohydrates and proteins in our bodies. Carbohydrates like starch are broken down into soluble sugars like glucose. Glucose will either be used immediately for aerobic respiration, where the chemical energy stored in glucose is released. If the body has excess glucose, then it will be stored in the form of glycogen in mainly our liver and muscle cells. Glycogen can be converted back to glucose when the body demands more energy. Proteins, when digested to amino acids, are used to build new proteins in our own bodies. For example, bodybuilders eat a lot of protein so they have enough amino acids to build their own muscular protein. Fats are broken down into fatty acids and glycerol, and our bodies use them mainly as an energy source. Fat is high in calories, meaning there is a lot of stored chemical energy available for our bodies to release. Depending on age and activity levels, dietary requirements will vary from individual to individual. In general, more active and younger individuals have a higher resting metabolism, meaning they burn more calories at rest. As you get older, the number of calories you need just at rest drops, and this is also the case if you have a very sedentary lifestyle. So, working out and exercising and building muscle can increase the amount of calories you need just to maintain normal bodily functions. You also need to know why we need the following vitamins and minerals. Iron is needed for the production of hemoglobin. Hemoglobin is found in your red blood cells, and it is this iron-containing molecule that allows your red blood cells to carry oxygen. Vitamin C is needed to help maintain healthy tissues in our bodies. Fiber is needed to add bulk to the digestive system. Effectively, it helps you to pass waste. Finally, water is an essential part to many of the body's functions and processes. Now to the health problems associated with a poor diet. If we eat too much sugar, this can lead to the following conditions: type 2 diabetes, obesity, and tooth decay. The reason excess sugar can lead to obesity is that the extra calories from sugar can be eventually stored as fat. Remember, if you don't exercise often, then you don't use as much energy, so any sugar not used can end up as fat. Obesity can lead to heart disease and circulatory disease. Effectively, fat and cholesterol can block your blood vessels if there is too much in your diet. Excess salt, specifically sodium, in your diet can lead to high blood pressure. A combination of high blood pressure and blocked arteries increases the risk of blood clots forming in your vessels, which can cause a stroke or a heart attack. This investigation measures how much energy is in different food. Normally, we burn different types of crisps in a classroom setting. The idea is that whichever crisp causes the greatest temperature increase in the water per gram has the greatest amount of chemical energy measured in joules per gram. This also means that that crisp has the most calories per gram. This video won't go through the method, but will instead go through the science behind the investigation and common questions they can ask on this investigation. Calories on food labels essentially describe how much energy is in that food. It takes roughly 4.2 joules of energy to raise the temperature of 1 gram of water by 1 degree Celsius, which is what scientists call the specific heat capacity of water. 1 centimeter cubed of water is equal to 1 gram. Luckily, all you need to do is plug the numbers from a question into this equation, which is given to you in the exam, and you do not need to worry about calories or the specific heat capacity of water. For example, calculate the energy per gram released by the Mr. Prick's special crispiest crisps of crisps using the following data. We have 20 centimeter cubed of water, so that means 20 grams of water, multiplied by how much the temperature increased, which is the final temperature, 67 degrees Celsius, minus the initial starting temperature of 20 degrees Celsius. So we plug in 47 degrees Celsius, followed by 4.2 joules per gram, which is the specific heat capacity of water, remember, and we divide this all by the mass of the food sample, which happens to be 1.5 grams. This gives us an answer of 2,632 joules per gram of energy in the crisp. It is important to note that the biggest temperature increase doesn't necessarily mean the most energy per gram, as you need to consider what mass of crisp you were burning in the first place. Burning a bigger crisp with more mass might give you the biggest temperature rise, but when you then divide it by its mass, you get that joules per gram ratio, which is the important number. Why some crisps have more calories in them compared to other crisps is all mainly down to fat content. Fat has a lot of chemical energy compared to carbohydrates and protein, so the crisps with more fat are going to be more energy dense and therefore have the most chemical energy per gram. You might also need to be able to identify the variables and where sources of error might come in when getting energy values. So the type of crisp is the independent variable. The dependent variable is measuring the energy content in the food per gram. The control variables are the amount of water we use, and we try to keep the distance between the burning food and the boiling tube the same. When we think of sources of error and inaccuracy, the key things are that it is difficult to burn the entire crisp, and often some of the crisp will fall off the mounting needle, meaning you may get a lower temperature increase than expected. This experiment's other main problem is that not all the heat energy from the crisp is transferred to the water, and you may get systematic errors where the food is not held at the same distance from the boiling tube whilst it is burning. We get a lot of heat energy also lost to the environment. In the food industry, they use a bomb calorimeter to measure the calories in the food. A bomb calorimeter has the food contained in a vessel, which can be remotely ignited. The vessel is surrounded by water, which is insulated and has a temperature probe inside. This means nearly all the heat energy released from the food is transferred into the surrounding water, and we get a measurement that is closer to the true value. In a class setting, you can use a cup calorimeter, which is a polystyrene cup that is insulated, but that is not suitable for the burning food experiment, but it is more suitable when measuring the energy released during a chemical reaction within a solution. We need to know how to produce basic drawings of a red blood cell and a white blood cell known as a phagocyte. A red blood cell has no nucleus, so we only need to label the cell membrane. The red blood cell has a biconcave shape. I like to think that from the side you see two caves. A tricky question from a past paper following the old specification asked why a red blood cell appeared paler at the centre when looking at it through a light microscope. You can have either answered it had no nucleus, or that it is thinner at the centre due to the biconcave shape, the more light passes through the centre. Handily, this shape provides a larger surface area for oxygen absorption, and they are flexible and small so they can fit through narrow blood capillaries. Finally, the red blood cells contain hemoglobin, which is an iron-containing molecule that enables a red blood cell to carry oxygen. Phagocytes have a much more irregular shape, and you'll need to be able to label the nucleus, cytoplasm, and the cell membrane. Phagocytes are a type of white blood cell, and they fight pathogens like bacteria and fungi. For example, they can engulf bacterial cells and then break down the bacteria using enzymes in a process called phagocytosis. The blood has four main parts: the red blood cells, which we have already mentioned, white blood cells like phagocytes, which help defend our body against pathogens, platelets, which are required for clotting blood. For example, if you have a cut on your finger, the platelets will clot and seal the wound and stop the bleeding. Finally, the plasma, which is made mostly of water, carries carbon dioxide, soluble food like glucose, urea, which is a waste product that will end up in urine, hormones like insulin, and finally helps to distribute heat around the body. When you get hot, your skin turns red, as blood is diverted to the surface so heat can be transferred to the environment more easily and help the body to cool down. In the neck, the heart is made of muscle, and the muscles contract to pump blood around the body. As it is made of muscle tissues, and therefore cells, the heart itself needs a blood supply, so oxygen, glucose, and other nutrients can be delivered via the coronary blood vessels. These can be seen on the outside of the heart. An important rule to remember before we label the heart is that arteries carry blood away from the heart, and veins carry blood back to the heart from the organs. These are all the labels you must learn for your exam regarding the heart. And remember, when looking at a diagram, you must remember that the right side appears on the left side of the paper. Just flip the paper and hold it against your heart, and you can identify which side is left and which is right if you are not sure. The heart has 4 chambers. The top 2 chambers, closer to your head, are called the right atrium and the left atrium. The bottom 2 chambers are called the right ventricle and the left ventricle. Between the right atrium and right ventricle is the tricuspid valve, which has 3 flaps. Between the left atrium and the left ventricle is the bicuspid valve, which has 2 flaps. A good way to remember which side the valves are on is it is right to tricuspid, and left behind, so bicuspid. The vein coming from the body and into the right atrium is called the vena cava. The artery leaving the right ventricle is called the pulmonary artery, and it has a semilunar valve. The vein coming back to the left atrium is called the pulmonary vein. The artery leaving the left ventricle is called the aorta and has a semilunar valve too. The heart is split in the middle, so that the blood in the right side of the heart does not mix with the blood in the left side of the heart. The right side only has deoxygenated blood, which contains red blood cells that have dropped off oxygen to cells in the body and now need to pick up more oxygen in the lungs, specifically the alveoli. The left side of the heart only holds oxygenated blood, which is red blood cells that have just come from the lungs, specifically the alveoli, and now need to deliver the oxygen to the cells in our bodies. The double circulatory system refers to 2 systems. One system for the lungs, where red blood cells need to perform gas exchange with the inhaled atmosphere. The biological term for this circuit is the pulmonary circuit. The second system is for the organs and the body, where oxygenated blood is delivered to the cells and organs, and deoxygenated blood is returned to the heart. The biological term for this system is the systemic circuit. Let us take a red blood cell that is deoxygenated and has just come from the tip of your finger and needs to get back to the lungs to pick up more oxygen. In this diagram, any vessels that are blue carry deoxygenated blood, and any red vessels carry oxygenated blood. So the red blood cell travels through the vena cava, which is the largest vein in our body, and into the right atrium. The right atrium walls will then contract and force blood into the right ventricle. The right ventricle walls will then contract, forcing blood up the pulmonary artery. Remember, all arteries carry blood away from the heart, and the name artery has nothing to do with whether they are carrying oxygenated blood or not. The tricuspid valve between the right atrium and the right ventricle stops the blood from flowing back into the right atrium. Valves stop backflow, meaning they only let blood travel in one direction. The tricuspid valve shape means that when the blood pushes against the flaps, they shut and close the path between the right atrium and right ventricle. The same is true of the semilunar valve of the pulmonary artery. It stops the backflow of blood so that all the blood from the right ventricle travels up the pulmonary artery, where it splits to either the right or left lung. Once the red blood cell has picked up some more oxygen, it returns to the left side of the heart via the pulmonary vein. Remember that all veins take blood back to the heart, and again has nothing to do whether it's deoxygenated or not. The pulmonary vein takes blood into the left atrium. The pulmonary circuit is now complete. The left atrium walls then contract to force blood into the left ventricle. The left ventricle walls will then contract to force blood up the aorta to the rest of the body. Please note that the left ventricle walls are much thicker than the right ventricle walls. This is because the left ventricle must apply more pressure to the blood, because the blood must travel further to the far reaches of the body like the head and limbs, for example. The right ventricle only must pump blood to the lungs, so the right ventricle does not need to produce as much pressure, hence the thinner walls. When the left ventricle contracts, the blood cannot flow back into the left atrium because the bicuspid valve flaps close shut, preventing backflow. The same is true of the semilunar valve in the aorta. It will flap shut as well if any blood tries to flow back into the left ventricle. The red blood cell will now deliver the oxygen to the organs and cells and finally return to the vena cava. The systemic circuit is now complete, and we can also say the red blood cell has completed its journey of the double circulatory system of the body. We need to know the structural differences and reasons behind them between the arteries, veins, and capillaries. Arteries carry blood away from the heart, and except for the pulmonary artery, it is always oxygenated blood. The blood just leaving the heart is always at a higher pressure, as it is closer to the pumping action of the heart. This means the structure of the arteries must cope with the great changes in blood pressure. Arteries have a tough outer coat and thicker wall compared to the veins and capillaries, and they generally contain the thickest layer of smooth muscle. The layer of smooth muscle is very elastic, so it can increase in volume as the ventricles contract and eject blood into the arteries at a high pressure. When the ventricles relax in between contracting, as they fill with blood, the elastic smooth muscle can spring back in the arteries, reduce the volume of the lumen, and maintain a high blood pressure to keep the blood moving. The inner wall of the artery is called the endothelium and helps to keep blood flowing, and it can release substances into the blood to regulate how fluid the blood is, and much more. The actual passageway through the artery is called the lumen. In arteries, the lumen is relatively smaller compared to what we see in veins, and the reason why the lumen is small in the artery is so it can maintain the pressure. Veins share many of the same features as the artery, but with some key differences. They carry blood back to the heart, and except for the pulmonary vein, it is always deoxygenated. In general, veins contain blood that is at a lower pressure, and their structure reflects that. The wall thickness is thinner because the pressure is lower. The muscle layer is thinner compared to arteries, and the lumen is generally larger than what you see in arteries to help keep the blood flowing. The veins also contain valves to reduce backflow. For example, in your legs, the veins need to get blood to travel against gravity and back to the heart. All this with a much lower pressure, so the valves help to keep blood flowing back to the heart. Muscle contraction in the legs also helps to keep blood moving by applying pressure to the veins and squeezing the blood upwards. The capillaries are the junction between arteries and veins. They have a very small lumen, and the wall is made of only one layer of endothelium, so only one cell thick. This is to reduce the diffusion distance between the blood, the cells, and the tissues. Capillaries form an extensive network through the organs and tissues, and this ensures that cells are always near a capillary so they can receive substances via diffusion or remove substances into the blood via diffusion. The risk factors for cardiovascular disease are as follows. If someone has too much fat in their diet, they may find they have high blood cholesterol and more fat deposited in their blood vessels. If someone consumes too much salt, this can lead to them having a higher blood pressure. Other risk factors include smoking, genetics, where their family has a history of cardiovascular disease, and if they do not exercise often. An atheroma is a buildup of fatty deposits in an artery. This is a particular issue if it occurs in the coronary arteries. As the fatty deposits build up, they narrow the lumen of the coronary arteries. This restricts blood flow, which means cells beyond the narrowing won't be receiving enough oxygen. This can cause heart pain, which doctors would call angina, and would be a warning sign of cardiovascular disease. Atheromas can also rupture, especially if the person has a higher blood pressure. This causes the body to try and clot the ruptured part. The platelets that clot the blood make a blockage in the coronary vessel. Now the heart muscle tissues beyond that blockage are receiving no oxygen and the cells start to die. This is what we would call a heart attack. If a clot were to block a vessel supplying the brain, then this would cause a stroke. Treatments for cardiovascular disease have their pros and cons. Statins have been shown to lower blood cholesterol and is an effective treatment to help fight cardiovascular disease by reducing the risk of developing an atheroma. Statins are tablets, so they are just a daily medication that is certainly less risky than surgery. Statins do, however, have possible side effects, and a few examples would be headaches, diarrhea, and problems with the liver. Angioplasty is a type of surgery that places a balloon inside the coronary artery that has a blockage and is inflated to widen the artery's lumen. This removes the blockage as the fat is pressed outwards and blood flow improves. This method can be temporary, however, but doctors can also put in a stent, which is a wire mesh that helps to keep the blood vessel open for longer. Like with any surgery, however, there is a risk of further complications, like the artery can become even more damaged where the stent was inserted. The final treatment you need to be aware of is recommending the patient to make changes to diet and more exercise. Healthy lifestyle choices include stopping smoking, exercising often, eating healthier food and less salt. All these can help reduce the cholesterol in your blood and lower your blood pressure, thus reducing the risk of suffering from further symptoms of cardiovascular disease. WJEC suggests that the problem with this treatment is that an individual needs a high level of discipline, as these changes must be long-term if they are to have a positive effect. Some organisms can produce food by using the energy in sunlight in a process called photosynthesis. Plants, algae, and cyanobacteria are examples of organisms that can photosynthesize. You must learn the word equation for photosynthesis, where carbon dioxide reacts with water and with the input of light energy produces glucose and oxygen. Chlorophyll, a green pigment found in the chloroplasts of plants, enables plants to absorb light energy and photosynthesize. It is also important that you know that photosynthesis is an enzyme-controlled reaction. Regarding the rate of photosynthesis, the following factors can affect how quickly a plant can photosynthesize. Temperature, carbon dioxide, and light intensity can all affect how much a plant can photosynthesize. Temperature could be too high or too low, and different species of plants will have different optimum temperatures for photosynthetic activity. If carbon dioxide levels are too low, then that will reduce the rate of photosynthesis, as will light intensity. We call these the limiting factors of photosynthesis. All three things can limit how much photosynthesis a plant can do. Focusing more on glucose and plants, they use the glucose they produce for many things. You must realize that plants make their own glucose, but they then must aerobically respire to release the energy in glucose to support other processes and activities within the plant. Plants can store energy by converting glucose into starch, so the plant can meet energy demands when photosynthetic activity is low. Glucose can also be converted into cellulose, which is what plant cell walls are made of. Plants can also use the glucose to make proteins and oils. Without photosynthesis, a plant can't make starch. Remember that glucose, a product of photosynthesis, can be converted into starch, and a plant will store starch in various places, including the leaf, where photosynthesis takes place. Using iodine, we can test for starch, but first you must prepare the leaf so the iodine can be absorbed into the plant cells and interact with the iodine. Step 1 is to boil the leaf in water to kill the plant cells and stop enzymic activity. Step 2 is to place the leaf in a boiling tube that contains ethanol. You can then place the boiling tube containing ethanol into the beaker containing boiling water. Ethanol boils at around 78 degrees Celsius, so we can turn the Bunsen burner off, as the heat energy left in the boiling water should be enough to boil the ethanol. Remember, ethanol is flammable. It's best not to have an open flame present. We boil the leaf in ethanol to remove the chlorophyll and remove the colour from the leaf. This is so we can more easily observe a colour change when we add iodine. Step 3 is to wash the leaf in cold water. This helps soften the leaf, and it can help to spread the leaf over a tile. The final step is to add iodine to the leaf and observe the colour change. Now we have a test for whether photosynthesis took place in a leaf or not. We can now investigate what happens if a leaf is exposed to different conditions and how that might affect photosynthesis and therefore starch production. Here is a plant that has had many of its leaves exposed to different conditions. It is important to note that this plant has variegated leaves, meaning the leaves have more than one colour. In this case, green and white. It is important that you realise that the white parts of the leaf on this plant do not contain chlorophyll. This will be really important later on. Leaf A has had nothing done to it. Leaf B is in a sealed glass vessel which contains 30 cm cubed of sodium hydroxide solution. You need to know that sodium hydroxide absorbs carbon dioxide, so this leaf will have no access to carbon dioxide. Leaf C will also be in a sealed glass container, but this one will contain 30 cm cubed of water. This acts like a control experiment to show that leaf B and C have access to the same volume of atmosphere, and it should prove that the sodium hydroxide is removing the carbon dioxide from the atmosphere and that the sealed container on its own is not affecting the leaf. Leaf D will have a strip of black tape covering a section of the leaf. It is important that before we start letting the plant photosynthesize, we need to be sure that the plant has no stored starch from any previous photosynthetic activity. So, we place the plant in a dark cupboard with no available light, so the plant uses up all its starch reserves. We call this destarching the plant. We place the destarched plant under a light source, so any starch being made now is new, and each leaf will now be photosynthesizing under their new respective conditions. After some time has passed, we can now test each leaf for the presence of starch. We get the following results where we show the before and after the starch test for each leaf. Leaf A shows that the green parts of the leaf have starch, but the white parts do not. This is because the white parts of the leaf have no chlorophyll, and therefore can't photosynthesize and make glucose, and therefore make starch. So this proves chlorophyll is needed for photosynthesis. Leaf B shows no positive result for starch because carbon dioxide is required for photosynthesis to occur. So no carbon dioxide, no photosynthesis, no glucose, therefore no starch. Leaf C is the same as leaf A, as this just again showed that the volume of atmosphere exposed to plant B was not affecting photosynthesis. So photosynthesis happened as normal. Eventually the leaf would be affected, of course, as the carbon dioxide would run out in the sealed container. But in this experiment, not enough time had passed. Leaf D shows a strip where the starch test is negative. This shows that the tape blocked the light and the leaf was not photosynthesizing where the tape was, and therefore not making any glucose, and therefore no starch. So this shows that light is needed for photosynthesis to occur. Finally, a more direct way to measure photosynthetic activity could be to use data loggers that can measure oxygen and carbon dioxide levels. You could seal a plant in a bag and measure the change in oxygen over time, and you should see a rise if the plant is photosynthesizing because oxygen is a byproduct of photosynthesis. So basically, the faster the oxygen is being made, the faster the plant is photosynthesizing. If you were to measure carbon dioxide levels, you should see carbon dioxide levels decrease over time as photosynthesis uses up carbon dioxide. And again, the quicker the plant is using up carbon dioxide, in theory, the faster it is photosynthesizing. Limiting factor graphs regarding photosynthesis are easy to interpret once you understand what a slope versus a horizontal line means from the plant's perspective. We will cover how to interpret these graphs and the specified practical, which is an investigation looking at how changing light intensity affects the rate of photosynthesis in pondweed. Here we have a plant inside a sealed bag. There is a data logger that's measuring the atmosphere inside the sealed bag. What it can do is it can measure the rate of photosynthesis by either measuring the oxygen produced over time or the carbon dioxide levels decreasing over time. Please notice that a second bag is sealing off the soil. This is to avoid microorganisms like bacteria or fungi respiring and affecting the oxygen and carbon dioxide levels, which may affect our data. We also have the option of placing sodium hydroxide solution with the plant if we wish to remove carbon dioxide from the atmosphere. But otherwise, we place water of equal volume in there. This is to try control the variable that is the volume of atmosphere available to the plant for each observation. Finally, for this example, we can also control the levels of carbon dioxide by using this gas canister. Starting with the carbon dioxide limiting factor graph, let's look at what happens to the plant's rate of photosynthesis when carbon dioxide levels are at 0 in the bag. As you would expect, the rate of photosynthesis is 0. We will gradually add more carbon dioxide into the bag and notice how the rate of photosynthesis is increasing. At this point, if we keep increasing carbon dioxide and the rate of photosynthesis doesn't level off, then carbon dioxide is limiting photosynthesis. The plant wants more carbon dioxide as it has the capacity to photosynthesize more. Remember this, slope equals carbon dioxide levels are too low. Carbon dioxide is a limiting factor. When the line levels off, this means the plant has reached its capacity to use carbon dioxide. No matter how much more carbon dioxide we add, the rate of photosynthesis is not going up. So we could say something else might be limiting the rate of photosynthesis. Remember, horizontal line equals carbon dioxide no longer limiting photosynthesis, so maybe another factor is. So we will increase the light intensity using this lighting rig. If we start by making it completely dark, we can see the rate of photosynthesis is now 0, even though there is plenty of carbon dioxide available. Now we increase the light intensity, and we can see that the rate of photosynthesis goes up. If the slope has not leveled off, then we know light intensity is the limiting factor for photosynthesis in this case, because the plant would be able to photosynthesize more if the light intensity was higher. So when the line is horizontal, this means that the plant has reached its capacity to utilize light energy, and increasing the light intensity more will not increase the rate of photosynthesis. So something else could be limiting photosynthesis. Now that we have carbon dioxide levels and light intensity at optimum levels, we can now control the temperature. This will produce a graph that is very similar to an enzyme activity versus temperature graph. The temperature where the rate of photosynthesis is optimum in this case is 24 degrees Celsius for this specific plant species. We can say that at the peak, the temperature is not the limiting rate of photosynthesis, but another factor might be. Notice that if we increase the temperature beyond the optimum temperature, we get a sudden drop in the rate, and this relates to the enzymes involved in photosynthesis denaturing. Therefore, we can say any temperature that isn't the optimum temperature is limiting the rate of photosynthesis. The specified practical that you have done in the lab, hopefully, looks at light intensity and the rate of photosynthesis in pondweed. We will just look at the variables, sources of error, and suggest improvements regarding this specified practical. The independent variable is the light intensity, which is controlled by placing a lamp at set distances from the pondweed. The dependent variable is counting the number of bubbles produced per minute, which reflects the rate of photosynthesis. The bubbles contain oxygen because oxygen is a byproduct of photosynthesis. More bubbles equals higher rate of photosynthesis. The control variables are using the same light source and the same plants for each distance. It is difficult to control the temperature in this experiment because the lamp is hot, and it could heat up the water when it is close to the pondweed and therefore affect the rate of photosynthesis. Another source of error is that the bubbles can vary in size, so the volume of oxygen produced could vary with each bubble. To avoid this problem, we could have the bubbles be bubbled into a gas syringe. A gas syringe can measure directly the volume of oxygen produced. We need to be able to label and describe the role of each part in this diagram of the leaf structure. You must imagine that this diagram is a cross-section of the leaf as if you're looking at it from the side. So, this is the top of the leaf, and this is the bottom of the leaf. Looking from the top, we have a waxy cuticle. This helps reduce water loss in the plant and allows light to pass through it. Below that is the upper epidermis, which is a single layer of cells. Next is the layer known as the palisade mesophyll layer. This layer is packed with vertically orientated palisade cells. Palisade cells are specialized for photosynthesis as they are full of chloroplasts. They are found in the upper layers of the leaf because they are more exposed to light, and it is more efficient to have them packed vertically as light can interact with more chloroplasts as it travels through the leaf and makes the most of the space available. Below the palisade mesophyll is the spongy mesophyll layer. The cells here are more spread out, and you have gaps available for gases like carbon dioxide to move throughout the leaf. Within the spongy mesophyll is the xylem and phloem. These are found in the leaf vein, and these travel throughout the plant. Xylem carries water from the roots to the leaves, and phloem carries sucrose, which is a type of sugar, from the leaves to other parts of the plant. Below the spongy mesophyll layer is the lower epidermis and a waxy cuticle, but notice it has gaps. These gaps are called stomata, which includes a pore called stoma and two cells encircling the stoma called guard cells. This is the site of gas exchange, where carbon dioxide, for example, can enter the leaf and oxygen can exit. Water can also be lost through the stomata in a process called transpiration. The guard cells can close and open to regulate the rate of transpiration and gas exchange. For example, if the plant needs to conserve water, the guard cells will close the stomata to reduce the rate of transpiration. In the cells topic, we covered the role water plays in supporting the plant cell structure. A hydrated plant will have a vacuole that is filled and that is pressing against the cell wall. We describe this as the cell being turgid. If the plant is not watered, then eventually the vacuole will start to shrivel up. This means the cell becomes flaccid as the outward pushing force is lost and the plant wilts. We also now know that water plays an important role in photosynthesis, as it is one of the reactants in the reaction. Without water, photosynthesis cannot happen. Roots absorb water, and the water is carried to other parts of the leaf by vessels called xylem. The water will contain dissolved mineral ions like potassium too. Remember that in the cells topic, we covered that root hair cells are specialised for water absorption by having a large surface area and contain lots of mitochondria. The root hair cells need lots of mitochondria because they use active transport to manipulate the water concentration within the root to encourage osmosis. Active transport moves mineral salts from the soil, where they are in a region of low concentration, into the root hair cell, where the mineral salts are in a region of high concentration. This requires energy because it is against the concentration gradient. Hence why the root hair cells have lots of mitochondria, which are aerobically respiring to provide the ATP to drive active transport. This means the root hair cells need a good supply of glucose and oxygen for active transport. The purpose of moving mineral salts into the root hair cell is so that the water concentration drops inside the root hair cell and the soil surrounding the root hair cell water concentration increases. Remember that dissolved salts lower the water concentration. Now osmosis can occur, where water moves down a concentration gradient. Water moves from where it is in a high concentration, which is the soil, into the root hair cell, which is a region of low water concentration, and the water diffuses across a selectively permeable membrane. Water then moves into the xylem, so water and some dissolved mineral ions can be transported to other parts of the plant. Remember, the xylems run through the roots, up the stem, and into the leaf veins to form a network that can help reach all the cells in the plant. Transpiration is the process where water exits the leaves via the stomata. This evaporation of water via the stomata helps to cool the leaf and regulate temperature. It also helps to pull water up the leaf, because water has a special chemical property that causes water molecules to be attracted to other water molecules. This means water follows water. So, when water leaves the stomata via transpiration, it helps to pull the water up the plant from the roots. We can indirectly measure the rate of transpiration using a potometer. We can't say that all the water we measure leaving the stomata is equal to what entered the roots, because some water is used in photosynthesis. What the potometer is great at doing is seeing what happens to the rate of transpiration when a plant is exposed to different conditions. Simply, when water evaporates from the stomata, the bubble travels further along the capillary tube towards the plant. So, the further the bubble moves in a set amount of time, the higher the rate of transpiration. These are the following conditions that affect the rate of transpiration. Higher temperatures increase the rate of transpiration because evaporation of water via the stomata is faster at higher temperatures, and the water diffuses more quickly out of the stomata and into the air. Higher wind speeds increase the rate of transpiration because the moving air removes water vapor near the stomata, which lowers the water concentration surrounding the stomata, so increases the concentration gradient. This causes more water to evaporate or diffuse into the local environment surrounding the stomata, thus increasing the rate of transpiration. Increased light intensity increases the rate of transpiration because it causes the leaf to open the stomata wider to try and maximize photosynthesis by increasing the uptake of carbon dioxide. This, however, also increases the rate of water loss that leaves the stomata. Higher humidity reduces the rate of transpiration because the concentration gradient of water inside and outside the stomata is less steep in humid conditions. This means the rate of diffusion of water out of the leaf is lower, so the rate of transpiration is lower too. In a lab, this can be achieved by placing a clear plastic bag over the leaf to trap the water that's leaving the leaf and increase the humidity in the local environment. Another factor that reduces the rate of transpiration is reduced surface area, and to achieve this, you can remove 1 or 2 leaves off the plant cutting, but you must remember to seal the now exposed stem with some Vaseline to reduce water loss. The reason why the transpiration rate is less is because there are less stomata, which means less water is now leaving the plant via transpiration. The plant makes glucose in the leaves via a process called photosynthesis. The plant then needs to transport the glucose to other parts of the plant that can't photosynthesize, so they can use it for aerobic respiration. The roots are underground, so there is almost no light there, and the root hair cells, for example, don't have chloroplasts because it would be a waste. Remember, root hair cells need glucose for aerobic respiration to drive active transport. The plant also needs to convert glucose into starch to store energy, and starch is stored in more places than the leaf. Potato plants, for example, send a lot of glucose into the tube underground, where it is converted to starch. However, the evidence suggests that in most plants, the glucose is converted into sucrose before it enters the phloem. We often refer to where the sucrose is made as the source, which is the leaf, of course, and what receives the sucrose as the sink. The scientific term for the transportation of sucrose via the phloem is called translocation. Farmers who grow crops will be very aware that without healthy soil, their crops will suffer. Farmers use fertilizer to add important minerals to the soil because plants need these minerals to build the complex molecules they need to survive. NPK fertilizers are used by farmers and gardeners because they contain nitrates, phosphates, and potassium compounds, hence the name NPK. Note, these are the elemental symbols for nitrogen, phosphorus, and potassium. Plants use nitrates to make amino acids for protein production. These are essential for cell growth. Phosphates are important because phosphorus is needed to make DNA and cell membranes. Potassium is needed for the enzymes involved in respiration and photosynthesis. You must learn what happens to a plant if they lack any of these 3 minerals. If a plant does not get enough nitrates, then this results in poor growth in general. If a plant does not get enough phosphates, then this results in poor root growth. If a plant does not get enough potassium, then this results in the leaves yellowing. The sun emits radiation, which is the main source of energy that supports life on Earth. Food chains visualize how the energy from the sun is transferred from organism to organism. We will use a simple example where we have the dandelion plant, which is consumed by the wild rabbit, and the wild rabbit is being consumed by the fox. At the bottom of every food chain is an organism that can make its own food, and we call this the producer. The dandelion uses solar energy to photosynthesize and produce glucose. However, not all the solar energy that gets to the dandelion plant is converted into chemical energy in the form of glucose. For now, let's just keep in mind that as we move up the food chain, energy is lost, and not all the energy is transferred from organism to organism. The first stage consumer is either a herbivore or an omnivore. Herbivores mainly feed on plant matter, and omnivores can feed on both plant and animal matter. In our case, the first stage consumer is the rabbit. The fox, a carnivore, which mainly consumes animal matter, is the second stage consumer in this food chain. Other terms can be used to describe food chains. For example, each stage can be described as a trophic level, and instead of a first, second, and third stage consumer and so on, we use the terms primary consumer, secondary consumer, tertiary consumer, and even a quaternary consumer. Here are some examples of food webs, which are very similar to food chains, but they can also show how more than one organism may rely on a food source. But they can be labelled exactly the same way. You just count how many organisms are in one chain within the web. You have the producer, first stage consumer, second stage consumer, third stage consumer, and fourth stage consumer. Or we can use the terms producer, primary consumer, secondary consumer, tertiary consumer, and quaternary consumer. To help us understand how energy is lost as we move up the food chain, we'll use some oversimplified numbers and some made-up energy values. Let's imagine that the dandelion plant has 1,000 kilojoules of solar energy reaching it in spring and summer, but it will only be able to convert a small percentage of that solar energy that reaches it for glucose production via photosynthesis. And one reason for this being is that simply photosynthesis is not 100% efficient. So for now, let's just say that 100 kilojoules is converted into glucose. The rabbit won't get 100 kilojoules from that dandelion when it eats it. The rabbit will probably only consume certain parts of the plant and is really only after whatever stored chemical energy the plant has to offer. The plant, however, did not use all that 100 kilojoules of glucose just for making and storing chemical energy in the form of starch, etc. The plant will use some of that glucose for respiration, and energy is lost as heat during this process. The plant would have probably produced during the spring and summer petals and seeds, and waste products that the rabbit won't have eaten. The rabbit in this case receives 10 kilojoules of energy from the dandelion. When the fox consumes the rabbit, it can't digest every single part of the rabbit. The rabbit will not have used all the 10 kilojoules of energy it received from the dandelion for producing muscle tissue, which includes repairing cells and growing new cells and tissues. It too will have respired and produced heat energy, which can't be eaten. You can't eat heat. The rabbit will also have produced waste products that again is energy lost and not passed on to the fox. Also, the rabbit will use some of that 10 kilojoules of energy that it got from the plant for movement and reproduction. And again, this energy is not available for the fox. So the fox only receives 1 kilojoule of energy from consuming the rabbit. Hopefully you can start to see why food chains in any habitat can't support endless food chains with carnivores eating carnivores eating carnivores and so on because of the energy losses. You also need to be aware that although at each stage energy is lost via uneaten parts of prey and waste, decomposers such as bacteria and fungi can consume that lost chemical energy for their own growth and reproduction. Another great way to visualize energy being transferred between organisms is using pyramids of numbers and pyramids of biomass. Pyramids of biomass always will have the traditional pyramid shape. Take a food chain that has an oak tree as the producer. The oak tree's biomass is 15,000 kilograms. As we move up the food chain, we observe a drop in biomass at each stage as energy is lost at each stage. The caterpillar, the primary consumer, in this case, is 150 kilograms worth of biomass. Obviously, this is thousands of individual caterpillars. The blue tits that consume the caterpillars have a total biomass of 5 kilograms. This again is quite a few individual blue tits. And finally, at the top of the food chain is the sparrowhawk, and that has a total biomass of 1 kilogram. And this won't be that many individual sparrowhawks. If we now use the same food chain but construct a pyramid of numbers, we don't always get the traditional shape, because the number represents the number of individuals of that species. So there is only one tree in this food chain. 5,000 caterpillars, 50 blue tits, 2 sparrowhawks. You need to be able to label and construct your own pyramids of numbers and your own pyramids of biomass for your exam. Finally, a higher tier skill that you need is how to calculate the efficiency of energy transfers between trophic levels. Simply, to calculate the efficiency, you divide the later stage in the food chain by the earlier stage and then multiply by 100%. If you have a bigger number on the bottom, the denominator, then you know you are going in the right direction. Also, you should never get an answer bigger than 100%. If you do, you have made a mistake. Here is an example. If you have 250 kilojoules of energy entering a deer, and then 5 kilojoules is passed on to the wolf when it consumes the deer, we put the later stage number on the top, the numerator, and divide it by the earlier stage, the deer, which is 250 kilojoules, and then multiply that by 100% to get an answer of 2%. The human population on Earth is set to continue to rise, which means humans will need more space for agriculture, housing, and industry. This extra space that is needed means wildlife is affected, as humans spread further into undisturbed habitats. Government agencies that overlook development projects into new areas must gather data to assess the effects the development may have on wildlife, taking a specific focus on endangered species. Biologists will collect data on how many endangered species are in an area and whether a new development may affect their food, water, and sources of shelter. The worst case scenario is that the development affects an endangered species to the point it becomes extinct. This means no individuals of that organism exist anywhere on Earth, and they have all died. Based on the data collected, a decision is made whether a development can be approved or not. Therefore, the government agencies have a responsibility to monitor, improve, and protect the environment whilst balancing human needs. Food production for human needs is provided by farming. Farming is what allowed humans in the first place to grow in such numbers, because farming methods can produce a lot of food for the space they use. Yield refers to how much produce a farmer makes. More crops, more yield. More meat, more yields. Farmers use fertilizers, pesticides, methods for controlling disease, and battery methods, all because they have the advantage of improving yield regarding crops and meat production. All these methods, however, do come with disadvantages. Fertilizer and pesticides have chemicals that can be washed away into waterways, which can have negative effects on organisms. When a farmer spreads fertilizer, there are guidelines and laws regarding when to do it and how much to use. If fertilizers enter the waterways, it can cause the dissolved oxygen in the water to be reduced, which in turn can kill off a lot of aquatic organisms and produce a dead zone. Here is the sequence of events that lead to this. Fertilizers and untreated sewage enter the waterways because excess rain and bad timing of fertilizer application causes nutrients to flood into the water. This causes a big increase in nutrients, including nitrates, phosphates, and potassium. The surface plants, and specifically surface algae, experience rapid growth, utilizing the abundant availability of nutrients. The algae specifically creates a layer on the surface, which blocks the light from reaching underwater. Plants that live underwater can no longer photosynthesize effectively and start to die, and therefore they are not releasing oxygen into the water. This is just the beginning of the dissolved oxygen levels dropping in the water. The algae and plants eventually use up all the nutrients and die off. Remember, this is a random increase in nutrients, and the waterway in this specific location can't support that high number of plants for an indefinite amount of time. The microbes in the water start to decompose the now dead plants and algae and break them down, and the microbes begin to reproduce and multiply. They use oxygen via aerobic respiration to do this, which causes the dissolved oxygen levels to decrease even further. The dissolved oxygen levels are now so low, animals start to suffocate and die. This area is now described as a dead zone. The scientific name for this process is called eutrophication. Farmers use antibiotics to reduce the incidence of disease, because disease would reduce the output that animal would have for things like meat, eggs, and milk. The disadvantages of using antibiotics is that if you use too much, they can be found in the actual meat product itself and can lead to an increased level of bacteria that are resistant to antibiotics. This is when bacteria who are more resistant to the antibiotics survive and can produce more bacteria that are resistant to antibiotics. The worst case scenario is that we're left with bacteria that can cause infections and the antibiotics no longer work. Battery methods involves having animals living in very close proximity to each other to maximize the space available to the farmer. The aim being is to get as much animal produce per square meter as possible. These methods can have negative impacts on animal welfare, and this could also mean the animal is not treated humanely and is suffering. We will focus on the process known as bioaccumulation and look at two specific examples where mercury, a heavy metal, and DDT, a pesticide, can enter food chains and can be toxic and cause reduced fertility and even death to the organisms at the top of the food chain. Let's take an industrial accident where mercury enters the coastal waters and begins to pollute the area. Let's use some simplified numbers and say that the mercury levels in each individual phytoplankton which have absorbed the mercury and are at the bottom of this food chain is currently at around 0.1 parts per million. This may not seem like much, but the US Food and Drug Administration recommend avoid eating fish that contain mercury levels above 0.46 parts per million. Shrimp then consume the phytoplankton. And as one shrimp will consume many phytoplankton, those shrimps will accumulate or build up more mercury in their tissues. So, one shrimp now contains mercury levels of 0.3 parts per million. Mackerel then eat the shrimp. And again, mackerel rely on eating many shrimps to survive, and the mercury continues to accumulate. Now, an individual mackerel contains mercury levels of 0.6 parts per million. Next, the tuna feed on the mackerel, and again, they eat many mackerel to survive. And we see mercury levels reach 1 part per million in an individual tuna fish. A great white shark then eats many tuna fish, and the mercury levels in the shark reach 2 parts per million. If people eat too many fish that contain high levels of mercury, they can suffer from mercury poisoning, which can affect the person's nervous system and damage their organs. In fact, some people eat shark fins, for example, and unknowingly could be consuming far too much mercury. DDT was a pesticide used to kill mosquitoes to reduce the spread of malaria and insects that fed on agricultural crops. It was very effective at killing pests, but there was evidence that it was killing untargeted insects that may be useful pollinators, and the effects of DDT were seen to be affecting animals higher up the food chain. Once DDT entered waterways and the food chains, it bioaccumulated, just in the same way as mercury did. Birds of prey, specifically those that fed on fish, had trouble absorbing calcium if they consumed high levels of DDT. This meant they had fertility issues. Specifically, the eggs they produced had thinner shells and broke before the chicks developed properly for hatching. This caused population crashes in the birds of prey. Indicator species questions in an exam are very much data interpretation questions. Effectively, biologists have gathered data on some species that thrive in certain pH or oxygen levels. We've previously mentioned how untreated sewage and fertilizer runoff can lower oxygen levels, so you might need to link why we see a high number of bloodworms, which thrive in low oxygen conditions, being found near the overflow sewage pipe. You will always be provided with information about what species does well in what conditions in the question. Another example of an indicator species that can be used to determine air quality is lichen. Lichen grows on bark, but is very sensitive to sulfur dioxide and will not thrive if there are high levels of sulfur dioxide present in the atmosphere. In this example, we can use the data and our knowledge and show that as we move further from the coal station, we can see more lichen growing on tree bark. Lichen is less abundant near the coal station because the sulfur dioxide levels are higher near the coal station. Microorganisms, such as bacteria and fungi, play an important role in breaking down waste matter produced by other organisms and dead organisms. This process of decomposition, when oxygen is available to the microorganism, release carbon dioxide into the atmosphere by aerobic respiration. The microorganisms do this so they can grow and reproduce. Decomposition is important because it returns materials that the once living organism took from the environment, so other organisms can use them. When decomposition is reduced, it can lead to the eventual formation of fossil fuels such as coal, oil, and gas. Conditions that lead to this vary, but essentially, low oxygen environments and pressure from above form fossil fuels. And of course, you need lots of time. These fossil fuels are known as hydrocarbons, and they are made of carbon, which has been taken from the atmosphere and stored in the ground. Fossil fuels are a great source of chemical energy, and we burn fossil fuels in a process called combustion, which releases the energy, but also the carbon dioxide, back into the atmosphere. The carbon cycle describes how carbon is incorporated into living organisms and then released again. Starting with plants, they photosynthesize, and carbon is captured from the atmosphere and converted into glucose, and then converted into other products that all contain carbon atoms. Organisms then consume the plants, and the carbon becomes incorporated into their cells and tissues. Carbon is found in carbohydrates, fats, and proteins, and this is the general way carbon is incorporated into life on Earth. Carbon is released back into the atmosphere by many routes, the main process being when living things respire, they release carbon dioxide as a waste product. Microorganisms that break down and decompose waste material and dead organisms release carbon dioxide as they respire. Finally, combustion, where carbon molecules are reacted with oxygen and release carbon dioxide back into the atmosphere. Some of this is natural, where combustion occurs during wildfires, for example. Human activities, such as burning fossil fuels for producing electric and powering cars, also release carbon dioxide back into the environment via combustion. Anyone who grows plants or vegetables knows how important it is to have healthy soil that is full of nutrients. Crops, for example, absorb lots of nitrates, phosphates, and potassium compounds out of the soil and incorporate them into their plant tissues. When we harvest that crop, most of those nutrients are removed from the field or vegetable patch. If you were to continue to do this, you will deplete the soil of nutrients and it won't grow healthy crops again. So farmers adopt many techniques to restore the nutrients in the soil after harvesting. They can add artificial fertilizers, which add phosphates, nitrates, and potassium compounds back into the soil. They can add manure, which is full of nutrients that can be decomposed and the nutrients can be returned to the soil. Finally, farmers can use certain crops that help return nitrates back to the soil. Gardeners will use compost, which is decomposed organic matter that is full of nutrients that can be returned to the soil and used by future plants. Microorganisms, such as bacteria and fungi, play an important role in making compost. Only organic matter goes in the compost pile, and the aim is to try break down the organic molecules into soluble molecules as quickly as possible. It takes a good 2 years to make good compost, but you can speed up the process. These factors affect how quickly microorganisms can decompose organic matter: temperature, oxygen levels, pH, moisture levels, presence of heavy metal pollutants, and the types of organic matter that you put in the compost pile all affect the enzyme action of bacteria and fungi. When trying to encourage fast decomposition in a compost pile, I personally would turn the pile every week to introduce more fresh oxygen into the pile. The microorganisms need to be aerobically respiring, as it is a faster process and breaks down organic matter quicker. I had a thermometer measuring the temperature, and I was aiming to keep the pile above 40 degrees Celsius, but would often have periods of the temperature being above 70 degrees Celsius. The reason compost gets so hot is because the microorganisms release heat as they aerobically respire, and that causes the temperature to rise because it is trapped in the center of the pile. You might think that 70 degrees Celsius would kill the bacteria, but thermophilic, meaning heat-loving bacteria, thrive in these conditions, and it handily kills plant pathogens that could infect your soil. pH can be affected by the ingredients you add to the compost pile. I would be careful not to over-add citrus fruits to the pile and make it too acidic, and mixing the pile helps to dilute the acid within the pile. I would not add anything inorganic to the pile because it won't break down, and of course, heavy metals such as copper inhibits microbial activity, so they don't go in either. Landfill sites are a problem because if lots of organic matter is sent there, you get poor decomposition, which ends up releasing more methane, a more potent greenhouse gas compared to carbon dioxide, into the atmosphere. Landfill sites are contaminated with heavy metals, and you can get areas of low oxygen, which leads to anaerobic respiration, which doesn't break down organic matter as efficiently, and it smells bad. A healthy compost pile should never smell bad. The nitrogen cycle shows us how nitrogen moves between the atmosphere, soil, and organisms. Nitrogen gas in the atmosphere cannot be used by the plants, but luckily there are bacteria that can convert the nitrogen gas into nitrates, which can be absorbed by the plants because they are soluble ions. Stage 1 of the nitrogen cycle is fixation, when nitrogen is converted into nitrates and fixed into the soil. Nitrogen-fixing bacteria, which live in the soil, can convert nitrogen gas into nitrates, but you need lots of oxygen in the soil for this to happen effectively. So, farmers can plough the field to turn the soil and reintroduce oxygen if needed. They also want to make sure the field isn't waterlogged, because that can also reduce oxygen levels. The worst case scenario is that there are denitrifying bacteria that can convert nitrates back to nitrogen gas, but these bacteria are mostly active in waterlogged and unploughed fields. Legumes, which are a type of crop, consisting of peas and bean plants, have root nodules that are special because they are home to many nitrifying bacteria. Farmers can rotate crops where they plant legumes in the soil to replenish the nitrate levels in the soil, instead of planting the same crop over and over again and depleting the soil of nitrates. Lightning can also convert nitrogen gas into nitrates, mainly because of the high temperature produced by the lightning bolt, breaking the nitrogen gas molecule apart, and the nitrogen atom is then absorbed into the soil and converted into nitrates. Industrial processes, such as the Haber process, also fixes nitrogen by converting nitrogen gas into ammonia. Stage 2 of the nitrogen cycle is when the nitrates are absorbed by the plants via the roots and the nitrogen is incorporated into the plants. The plants use the nitrogen to make proteins, for example. Stage 3 of the nitrogen cycle is when animals consume the plants and digest the plant proteins into amino acids and build their own animal proteins which contain nitrogen. Some of that nitrogen, however, ends up as waste in the form of urea, which is found in urine. The urea enters the soil and can be broken down into ammonia, and then the ammonia can be converted into nitrates. Some bacteria in the soil make the enzyme urease, which is what converts the urea into ammonia, and then ammonia can be converted into nitrates by nitrifying bacteria. Stage 4 is when animals die and get decomposed by microorganisms. The nitrogen in the animal proteins and tissues is converted into ammonia by the bacteria involved in decomposition. Nitrifying bacteria can convert that ammonia into nitrates. Stage 5 of the nitrogen cycle is when the nitrogen is returned to the atmosphere. Again, this is done by denitrifying bacteria found in the soil which thrive in waterlogged and low oxygen conditions. So, we have completed the triple award biology content.